Hey y'all, welcome back to my channel. My name is Jeffrey. In this video, I'm going to discuss with you guys translation and just a brief overview of everything you need to know. Translation, remember, is the production of protein. So if we just look back here, protein production is made from an mRNA transcript, and you're going to have a ribosome that is going to bind to that mRNA and make a polypeptide or protein with amino acids that are brought in from tRNAs. So what's going to happen here is you're going to have the mRNA here, right? And it's going to be bound by the small subunit of the ribosome. And the small subunit is going to bind to a recognition site on the mRNA. It's going to scan downstream of the mRNA until it finds the start codon. And the start codon is universal. It's always AUG. In this instance where it actually scans and it reaches this this codon, what's going to happen is that's going to initiate the recruitment of the large subunit, and now you have your finished translational machinery. So that's going to be the initiation of translation. So then for elongation, essentially what's going to happen is that you're going to have tRNAs, and these tRNAs are going to be carrying amino acids on them, and they're going to go into the EPAs, the different sites, right? Remember, the large subunit always has 3 sites. It has the exit site, the peptidyl site, and the amino site. So during elongation, what's going to happen is you're going to have that tRNA. It's going to go into that A site. And remember, in the P site, there's already a tRNA here with a polypeptide, right? And what's going to happen is that you're going to have this tRNA, it's going to have the amino acid on it, and you're going to expend some energy, usually some GTP here, to form a peptidyl bond between the incoming amino acid along with the polypeptide chain. And that's going to actually transfer this over. So essentially, you're going to have the polypeptide now in the A site. And then what's going to happen is you're going to expend some more energy, and you're going to kick this tRNA out into the E site where it exits. And then this tRNA in the A site is going to be translocated into the P site. So, and then you repeat that process over and over again until you reach the stop codon or some other way of transcriptional termination. Okay? So that's just the brief overview of the 3 stages of translation: the initiation stage, the elongation stage, and the termination stage. So now for prokaryotes, there are going to be a couple of differences, right? So for prokaryotes, you have this thing called F-MET, which is also called informal methionine. It's different from eukaryotes, which is just methionine carried on a special tRNA. So for prokaryotes, what's going to happen is, once again, you're going to have that small subunit. It binds to this recognition site, and this recognition site is going to be called the Shine-Dargano sequence. And then it's going to go downstream once again until it reaches the AUG site. And this sequence is actually just 5 to 10 units upstream of the start codon. So now what happens after it reaches that AUG, you're going to have initiation factors, and these just help with the process of translation. So your IF2 GTP is going to bring in your informal methionine to your P site. And the reason why I say P is because, this is the initiation. You don't have anything in the P site because this is the first amino acid you're going to have. So it's going to bring that to the P site, and then your GTP is going to hydrolyze. So this GTP is going to be hydrolyzed, and that is going to finish up your initiation stage, because this is going to bind the 50S subunit, which is the large subunit, onto that start codon. So then after that, your initiation factors just release, and now you have your finished translational machinery to go downstream and code for the rest of the polypeptide. Okay. Now you're also going to have this elongation factor, your EF2GTP, and this is going to actually bring your appropriate tRNAs to that A site, and then you're going to have your GTP once again hydrolyzed to actually deliver that that tRNA to the A site. Okay. So finally, to form that peptide bond, you're going to just need something called peptidyltransferase, and this, it makes sense because you have that peptidyl. It's self-explanatory in the name. So it catalyzes your peptide bond formation, and it transfers your amino acid from the P to the A site, like I was talking to you guys before. So P to A, right? And then finally, you're going to have elongation factor G, GTP, and it's going to translocate your A site tRNA to your P site by hydrolyzing your GTP, right? So that's just going to be your elongation stage. You're just going to keep repeating that over and over and over. Now, for eukaryotes, what's going to be different from this entire thing is just the fact that this EFG GTP is now called EF2 in EF2 GTP in eukaryotes. Okay, so just keep in mind all of these initiation and elongation factors because they are very important and you can be tested on them. And then finally, to terminate, you have a termination codon usually, and this is going to cause your ribosome to stall before it's going to be recognized by something called an RF or release factor. And then this is essentially just like a block, if you will. And then, so if we look back into our large subunit, remember that this is usually occupied by a tRNA. And if it's occupied by tRNA, your transcriptional machinery keeps going. But what's going to happen here is you're going to have this thing called a release factor. It's kind of just an empty block. It goes into the A site, no peptide bond forms. This ribosome is going to just kind of like stall, and then eventually it's just going to dislocate from the mRNA. So what's going to happen here with the release factor is that now you're going to have that peptidyltransferase, and it's going to cleave an ester bond between the tRNA and the polypeptide, and then it hydrolyzes a GTP to dissociate from the mRNA complex. All right, so that's going to be essentially everything you need to know about translation. Now, there are many, many drugs that affect different aspects of translation. So, for example, streptomycin is going to affect the initiation of transcription, which is going to bind to this 30S subunit. So streptomycin is going to bind to the 30S subunit. It prevents initiation completely. So you're not even forming a single peptide bond. You're not having the recruitment of even the large subunit, right? So that's going to be, very big in preventing certain bacterial infections, for example. And then for tetracycline, that's going to prevent A site access to the tRNA. For elongation, right? You have erythromycin that's going to block translocation. It's going to bind to the 50S subunit of the actual complete ribosome. So these are just there are many different drugs. I've actually compiled a list of all the drugs you really need to know that affect your protein synthesis, and I'm going to have that linked in the description below. All right. So let's just talk about after you form this entire polypeptide, right? So you now you have the entire translational machinery disassociated, and you're going to be left with a string of these amino acids, right? This polypeptide right here. This by itself is going to be called a zymogen. So zymogen is kind of a inactive protein, right? So for example, if you think about maybe like those glow sticks, remember when you were young and you played with them, remember that you had to crack them first, right? You had to kind of like break them in half and shake it up a little bit, and that activates the glow stick so that it actually works and achieves the effect that it's supposed to achieve, right? Before it wasn't glowing, and then after you cracked it, it starts glowing, right? So after you modified it, it glows. That same thing happens for protein. So when you first make a polypeptide, it's going to be called a zymogen. It's inactive, and you're going to activate it by cleaving off, certain parts of that polypeptide. You're going to have these things called kinases. Kinases basically just phosphorylate things, and phosphatases, they just add a phosphate group. Or no, sorry, they dephosphorylate, which is essentially the same thing as taking off a phosphate group, right? Okay, so those are the different types of proteins there are, and then you're going to have some things called glycosylation. These just help these proteins get recognized. So these are known as recognition signals. So glycosylation, there's glycosylation and O glycosylation. So your O-linked and your N-linked glycosylations. And O-linked is just going to be a carbohydrate attached to your OH of a serine or threonine, and this is only after the polypeptide reaches the Golgi body. N-linked is essentially where you have a carbohydrate or sugar attached to the nitrogen side of a asparagine amide, and this is going to happen in the endoplasmic reticulum. So that's just something to keep in mind. I don't think it's that important at this stage in your education, but it'll be neat to know about O and N-linked glycosylation. And just remember that glycosylation is just adding a sugar or a carbohydrate. And then you can also have this thing called lipid anchoring. Lipid anchoring is essentially just adding a farcinyl group to a RAS protein to the plasma membrane inner leaflet. So, is essentially what this does is you have your plasma membrane right here, and you have a lipid coming out of it as recognition, and this is just going to anchor that lipid to the plasma membrane. The final thing I'm going to talk about is just insulin C-Peptide. I'm sure you guys learned about it, but insulin and C-Peptide are going to be packaged together for secretion because one of them allows for the activation of the other. So C-peptide is essential for your proper insulin folding, right? So if you don't have C-peptide, your insulin is likely not going to work. So that's why for some insulin injections, or sometimes they just inject you with C-peptide because your insulin is actually produced fine. There are no mutations. It's just that you need the C-peptide for the proper folding to allow for it to achieve the desired function of insulin. So you have a signal sequence that's going to recruit your ribosome to attach your insulin polypeptide to your rough endoplasmic reticulum. It's going to be cleaved to form proinsulin, which is going to be the zymogen in this instance. It's going to be transported to the Golgi body and cleaved to form your insulin, which is going to be the active form with C-peptide. And then that's going to be secreted out of the cell via secretory vesicles. So I hope that this video has been helpful. I hope that you guys learned a thing or two about translation. Sorry if it's a very long video or my notes are not really written out, but everything I said was factually correct. So thank you. Have a great day.